Programming DNA may seem like a concept from a Hollywood sci-fi movie, but in reality, programming DNA is happening all the time in the field of synthetic biology. Synthetic biology is a strand of life science where scientists read, write, copy, and paste certain sequences of DNA to design innovations that can give humans a better quality of life. But this concept of programming DNA can seem pretty confusing. So let's get into the specifics. It can seem extremely confusing, but it isn't that hard to understand. Let's imagine that DNA is made from 4 types of blocks: green, red, blue, and yellow. These blocks make up DNA. They represent adenine, cytosine, guanine, and thymine. There are millions of these ACTGs for short, and certain sequences perform certain functions. How can we read DNA? What do these strands look like, and what functions can they perform? Well, we can tackle that first question by learning about a DNA sequencing machine. To make it simple to understand, we can imagine a DNA sequencing machine as a barcode scanner that scans DNA. Each and every DNA that gets scanned has a unique barcode. The barcode is read as millions of these ACTGs. This is how we read DNA. When picturing what strands look like, we can imagine a book just filled with the millions of characters. A, C, G, and T. Scientists can find the starting and ending point just like how we can find the starting and ending point of a statement. We can imagine a full strand as a highlight in a notebook. This is how we can visualize what strands look like. So, what functions do these strands perform? Well, like I said, ACTGs make up the DNA, and the DNA B sequences, well, they're leaders. They carry the information a cell needs to assemble protein and RNA molecules. Proteins are essential to any life because they act as receptors, transport molecules, and regularly protein for gene expression. Scientists can cut, copy, and paste certain sequences of DNA, or what we can visualize as these blocks, and combine them with other blocks to create a brand new innovation. So, this begs the question, what kind of innovations can we create? Well, we can make medicines more efficiently. Right now, diabetes is on the rise, with an estimated 463 million people who have it worldwide. With programming DNA, instead of getting insulin by killing farm animals, the traditional way of getting insulin, which is not even good for the environment because of the factories used in the process, we can instead program DNA to combine human insulin genes with the bacteria to create a medicine that is a fraction of the cost and more environmentally safe. And this is just one application for synthetic biology. The applications for synthetic biology are endless, and a new era of innovation is upon us. If you can imagine it, synthetic biology can create it. Thank you.